A fundamental challenge in biology has been the ability to understand how individual cells behave within complex heterogeneous tissues. Whether in healthy organs or disease states, cellular populations are rarely uniform. Cells of the same type can behave very differently depending on their environment, their activation state, or interaction with neighboring cells. So thankfully, powerful new tools have allowed us to characterize each of these individual cells, parse them into different subsets, and then measure their differential responses. This, of course, started with flow cytometry, the veritable workhorse in this space where these so-called clusters of differentiation are created from distinct protein patterns. As a simple example, one could distinguish between different cell types in a tumor microenvironment, tumor cells from stromal cells from lymphocytes. Further advances in single-cell multiomic technologies more recently have allowed us to dive far more deeply into the differential behavior of cells within these subtypes. For example, one could differentiate different T cells that are activated from those that have not been activated. And even more recently, we can now map them spatially within the context of each other and in their native tissue, allowing us to better understand the activity of these cells with reference to their location within, let's say, a complex tumor microenvironment. So intact spatial context of this rich multiomic technology. But in all of their value and power, these tools only allow scientists to infer the actual behavior of these cells, to make these mathematical guesses of what's actually occurring from all this very rich multiomic data at the single cell level. So this brings us to the next chapter in therapeutic discovery, which will come on the shoulders of being able to directly measure single cell function. Whether it's killing, secretion, or internalization, our ability to directly measure functional behavior at the single cell level will open up doors to a whole new powerful class of therapeutics that were previously uncharted.